Hello, this is my stop motion video on insulin cell signaling. The insulin, after being produced from beta cells within the pancreas, arrives at the hepatocyte cell within the liver after being signaled by the presence of glucose in the pancreas. The insulin binds to the insulin receptor, and in this case, it's a tyrosine kinase receptor. This binding halts the cell's release of glucose and triggers the uptake of glucose into the cell. The binding also initiates a signal cascade that stimulates the insulin receptor substrate that then goes on and binds with the phosphoinositide 3 kinase, or PI3K, activating it and producing PIP3. The PIP3 goes on to bind with a protein kinase B, which afterward can become phosphorylated. After protein kinase B's own phosphorylation, it then further phosphorylates with glycogen synthase kinase 3, or GSK, by giving up the phosphate groups it received from ATP and transferring them to GSK3. This phosphorylation, however, inhibits GSK3's activity. Without the activity of GSK3 halting glycogen synthesis is able to begin, with the final product being a molecule of glycogen after 6 of the glucose molecules taken in by the cell are bonded together to more efficiently and effectively store glucose over a long period of time. Further cellular responses caused by the insulin transduction pathway, on top of an increased glucose uptake and glycogen synthesis, is an increase in protein synthesis, an increase in DNA synthesis, an increase in amino acid uptake, as well as a decrease in apoptosis due to the limiting and halting of the cell's activities during the time that the insulin is bonded to the cell's receptors.